Hello everyone. Here we are to discuss on the topic of Western blotting. In my previous lecture, I have already discussed about the different techniques of blotting, and in detail we have discussed about Southern blotting. Now we get to know what is Western blotting. The Western blot is an analytical technique which is used to detect the specific proteins in a given sample of tissue, homogenate or extract, and it is a part of immunoblotting because an antibody is used to specifically detect its antigen. Western blot was mainly used for protein, and it was developed by George Stark by using different antibodies to locate the proteins. Western blotting is based on building antigen antibody protein complex through specific binding of antibodies to proteins which are immobilized on the membrane. It is followed by detecting the bound antibody through several detection methods. Here we are going to discuss the different steps of Western blotting, or we can also say we will discuss about the Western blotting workflow. The first step is the preparation of the sample which needs to be analyzed. Next, we need to carry out a process of gel electrophoresis. Then the transfer of the sample is done. After that, the blocking and antibody probing is done. The results is then detected by the different process of detection, and analysis of the results are done at the end. First step, that is the sample preparation. In this particular step, the proteins from all sources can be used for Western blotting, such as from single cell, whole tissues, extracellular matrices, biological fluids. The protein can be extracted from the sample in three distinct steps. First step is cell lysis. It is a physical method in which ultrasonication or homogenization can be done. If you are using a chemical method, you can use the detergents for causing the cell lysis. And if you are going for a biological method, then enzymatic digestion is the best method of choice. Next, the protein isolation from the rest of the cellular components is carried out by the process of centrifugation. DNase and other lysis buffers are used for the removal of cell organelles, nucleic acids, polysaccharides, etc. And further, the purification of protein is carried out. After the sample preparation is complete, we proceed further for the step of gel electrophoresis. Page gel electrophoresis, that is polyacrylamide gel electrophoresis, is mainly used for the separation of protein particles. Again, I am repeating, page gel electrophoresis, that is polyacrylamide. SDS, that is sodium dodecyl sulfate, is added to the gel and running buffer to denature the protein molecules, which will ensure that your proteins are separated solely on the basis of size and not on the 3D structure, the three-dimensional structure. The molecular weight markers are used to define the size of the proteins that run in a gel. Markers are usually composed of different proteins of known size, so that you can compare the markers, the known markers, with the unknown protein sample. You match the reading and you get to know the size of the unknown sample. As proteins are not directly visible in the gel must be stained. The proteins are usually stained with dyes such as blue, silver stain or deep purple. After the step of gel electrophoresis is complete, the protein contained in the gel is subjected to transfer. The next step is protein transfer. The protein are transferred from the gel to a solid support membrane such as nitrocellulose or polyvinylidene difluoride, PVDF. We have already used this in case of Southern blotting as well. The proteins transferred from the gels are immobilized in order to make it possible to detect the proteins on the membrane by using the specific antibodies. If it is immobilized, you can easily detect with combination of antibodies. It will bind to the antibodies. Electroblotting is mainly used, which uses the electric field to pull proteins from the gel onto the PVDF or nitrocellulose membrane. So, how the protein is transferred? By the use of electroblotting. The proteins move from within the gel onto the membrane while maintaining the organization that they had within the gel. I hope this step is clear to you. This is a diagrammatic representation of how the gel is taken out and the protein transfer takes place by the help of electroblotting. Here you can see we are applying the electroblotting technique. This is the cathode, this is the anode. Blotting stack on the cathode side of the gel, blotting stack on the anode side of the gel, and this is the gel which contains the protein. Okay, so now you can see here that it is transferred by electronic application, electrons transfer from gel. So, you can see the protein which were present here has been transferred to the blotting stack. Blocking and antibody probing is the next step. In Western blotting involves the immobilization of biomolecules on a membrane and blocking the spaces not already occupied by the proteins. As non-specific binding of antibodies to the membrane reduces the specificity and sensitivity of the assay. Two main classes of blocking agents are protein and non-ionic detergents, which are very commonly used for the purpose of Western blotting. Antibody probing. In this step, the Western blotting protocols usually uses a non-labeled primary antibody directed against the target protein and labeled secondary antibody directed against the constant region of the primary antibody. The secondary antibody serves not only as the carrier of the label but is also a mechanism to amplify the emitted signals, because the secondary antibodies are most often polyclonal and can bind primary antibody at different epitopes simultaneously. Why it can bind to the different epitopes? Because of the polyclonal nature. Alkaline phosphatase, that is abbreviated as AP, and the horseradish peroxidase, that is HRP, are the two most commonly used enzymes of protein detection using chromogenic substrates. This was also used in the case of ELISA. What are the other antibody probes used for protein detection? They are fluorophores, biotinylated secondary antibodies, and radioisotopes. How detection is carried out? Different detection systems are used in Western blotting based on fluorescence, chromogenic or radioisotopic detection. Chromogenic deals with the color, radioisotopic is for labeled probe, and fluorescence is the detection under UV radiation. Enzymatic detection methods require addition of a substrate that emits light when it reacts with an enzyme conjugated to a secondary antibody. Fluorescence based detection requires the excitation of fluorophore using a light source of a specific wavelength which can cause the light emission to happen, and as a result the fluorescence is given out and it is detected by the detector. This is a chemiluminescence example. Here you can see the ECl reagent is poured onto the protein and excitation and emission of the fluorescence is shown here. Okay. Side-eye conjugated secondary antibodies are there, and these are the primary antibodies. And this is the proteins on the membrane. This is fluorescence based detection, and this is chemiluminescence. How the analysis is carried out? The detection of the signals using either X-ray film scanners or a charge coupled device, that is CCD, any camera based imager, results in image containing visible protein bands. See, you can see here the protein bands. This is the standard plate which contains the known sample of the known molecular weight, and this is the sample plate 1, 2, 3 and 4. Okay, these are the markers. This is the plate for markers. The presence and the amount of protein of interest is estimated by visual analysis, and the size can be determined by comparison to a known molecular weight marker. See, this band is at the same position that of this band. So, this can be concluded that this particular sample number 2 is having a molecular weight of 76,000. Okay. What are the different applications of Western blotting? They also have a wide range of applications. They are very useful in the diagnosis of HIV by ELISA, which involves the Western blotting technique. Western blotting technique is also used to detect some forms of Lyme diseases. Western blotting technique is used in definitive test for BSc. Confirmatory test for hepatitis B involves Western blotting technique. This technique is used in the analysis of biomarkers such as hormones, growth factors and cytokines. And this technique is also employed in the gene expression studies. What are the limitations of Western blotting technique? This particular process is very delicate and time consuming. A minute imbalance at any level of the procedure can skew the results of entire process. It can also cause erroneous in bands or no bands due to insufficient transfer. That means at the transfer of protein step, if the transfer is not properly done, the bands will not be visible at the detection point. It should require well-trained techniques, and for this technique, specific skilled persons are required. Primary antibody availability is very crucial. It is just a semi-quantitative test, only estimation and not a precise measurement of the molecular weight of the protein is possible. With this, I conclude my presentation on this topic. The Western blotting is based on the specific interaction between antigen and antibody. It is widely used as diagnostic tool in medicine and as quality control measures in various industries. It is also used as an analytical tool in biomedical research for the detection and quantification of specific antigens or antibodies in a given sample. I hope by following my lecture you will have a clear picture of the Western blotting in your mind. Thank you for listening to me.